Now, the first type of epithelium we'll talk about is the simple squamous epithelium. Just based on the name alone, we know that it's one layer, simple, of flattened cells, squamous. And if it's an epithelium, it's going to be lining a space of some sort. So if you look here at our microscopy image on the right, you'll find that, okay, this is actually looking at tissue under the microscope. And if you want to find your epithelium, locate your space. And you can find here that there's a lot of spaces. Like all of this is a space, here's one, here's a space, which means that if these are all spaces, the epithelium is what would cover and line that space. So that means that this is the epithelial tissue that forms the boundaries of that space. Specifically, what we're looking at here is actually lung tissue, which is why you have large air-filled spaces. And the epithelium that lines these spaces is simple squamous, because if you look closely, they're very, very flat cells, almost indistinguishable, and they appear to be one layer, so you call that simple squamous epithelium. And you might wonder, okay, well, what's this stuff good for? Like, why do you want simple squamous epithelium in your lungs? Well, because of the fact that it's thin, it allows materials to diffuse and filter very rapidly. It also secretes lubricating substances and things called serous membrane that we'll get to in a future chapter. And some of the places you might want to find a simple squamous epithelium because of rapid diffusion would be things like your kidney glomeruli, where filtration occurs, the air sacs of your lungs, where you need rapid exchange of gases, the internal and external linings of your heart for lubrication and protection. You'd find this lining the inside of your blood vessels for rapid diffusion of nutrients, as well as your lymphatic vessels, again, for rapid diffusion of nutrients. So when you think of simple squamous epithelium, think of rapid exchange of material from your space deeper into the connective tissue down here. Now the next one here, we have simple cuboidal epithelium. So remember, if it's simple, one layer, cuboidal, cube-shaped cells. So if looking at our microscopy image on the right, we got to find our space first, right? And this one looks much more dense than the previous slide because you're looking at a denser organ. In this example is your kidneys. So if we want to find our epithelium, we got to find our spaces. So if you look closely, this is a space here, that's a space here, so are these up here. So if we find our space here, we should know that our epithelial cells line that space. So if you look at the cells that are lining or surrounding the space, you'd see, okay, they look cube-shaped, and as far as we can tell, there's one layer of cells, and you can actually see the basement membrane here that the cells attach to on their basal surface. And because it looks like cube-shaped one layer of cells, you'd say this is simple cuboidal. Now, simple cuboidal epithelial tissues are really good at secretion absorption. The cells are a little thicker than simple squamous, so they're slightly more protective. And the places you'd find simple cuboidal epithelium would be things like your kidney tubules, some of the secretory portions of smaller glands, as well as the surface of the ovaries. Now this one is simple columnar epithelium, and again, it's one layer of column-shaped cells. And you remember, you want to find your space when you're looking at a microscopy slide here. So here's our space. And if this is a space here, then you should find cells just next to it. And you can see here, okay, well, these cells look really long, and as far as we can tell, there's one row. So we know it's simple. And if you look at these cells, they don't appear to be stacked in terms of lots of layers. You'd see that here's their kind of basal boundary here. And based on the shape, they look columnar, so you'd say that this is simple columnar epithelium. Now, simple columnar epithelium is a single layer of tall cells with kind of oval nuclei. Now, this is really good at absorption, as well as secretion of mucus, enzymes, and other substances. There is a ciliated variety you find in some of the reproductive cells, like in the oviducts or ovarian tubes. But the non-ciliated types of simple columnar epithelium are what line most of your digestive tract. And the reason why you'd want a simple columnar epithelium along your digestive tract is that these cells are nice and long, which makes them more protective, but there's still one layer of cells, which means they're still okay at absorption. Now, they might not absorb as fast as simple squamous, but they're also more protective, which is important in your digestive tract versus your lungs. So this makes sense why you'd find a lot of simple columnar epithelium all along your digestive tract, or at least along most of it. You also find simple columnar epithelium in the gallbladder, some glands. You find a different variety in smaller bronchi, as well as the uterine tubes and portions of the uterus as well. So this is simple columnar epithelium. Remember, it's really good at absorption and secretion, but it's also more protective than the other simple epithelia, like simple cuboidal and simple squamous. So this one here is an atypical epithelium, and the reason why we say atypical is it doesn't really fit our classic definition of, how to classify epithelia based on the shape and number of layers. In fact, this one here is called pseudostratified ciliated columnar epithelium. And the reason why it's pseudostratified is that these cells appear to be stratified. If you look here, they almost look like they're stacked upon each other. But in reality, there's actually one layer of column-shaped cells. The problem is that when you cut through the tissue and you look at these tissues in 2 dimensions, it appears to look like the cells are stacked upon each other, but really they're just at angles, and so it's in fact a simple epithelium. So that's why we say it's pseudostratified, because it looks like it's stratified. Now, this one's really easy to identify in terms of these long cilia. The cilia here are located on the apical surface of these cells, and they actually aid in the propulsion of mucus out of your airway. And if you find these long cilia, basically you would know immediately this is pseudostratified ciliated columnar epithelium, because we really won't be going over other epithelia that appear like this with cilia. Now, you also find that in the epithelial tissue, there's these large cells here. They look like large spaces. In reality, these are actually just large, what we call goblet cells. And these goblet cells are unicellular mucus cells. And what they do is they basically create mucus and then secrete that mucus onto the apical surface. That way these cilia can wave within a nice mucus layer and also trap debris. Now, the stratified squamous epithelium you find in areas that need to resist abrasion. Think of your skin, or even your oral cavity, anal cavity, or vaginal cavity. Now, this is a thick membrane. It's stratified. If you look here, there's lots of cells. And you might look at the basal surface and go, hey, these look kind of cube-like, because the cells are sort of more rounded. However, remember, stratified epithelia are classified based on the shape of the cells at the apical surface, not the basal surface. So apically, you can see that these cells are more flattened because they almost look like they're very squished. You can see the nuclei here are squished. And so we would say that this is squamous because there's lots of layers. We say stratified squamous. Stratified squamous is not very good at absorption because these are way too many cell layers to absorb that well. But it is good at protection. So stratified squamous epithelia tissues you'd find in areas that need to protect and resist abrasion. So you find the non-keratinized forms in the moist linings of like your esophagus, mouth, vagina, as well as the anus, and the keratinized variety in the epidermis of your skin. So the outer layer of your skin is actually made of a keratinized stratified squamous epithelium. And we learned about in chapter 3 how keratin is a very tough protein that's involved with basically protection. Now, transitional epithelium is also one of those atypical epithelial tissues. Because if you look here, you almost would be inclined to call this stratified cuboidal. Because if these cells look like they're stacked on top of each other and they're kind of cube-shaped. Now, this one's unique because you only find it in the ureters and urinary bladder of your urinary tract. And this type of epithelium is really good at stretch. So by having lots of cells that are really large and pillowy, this epithelium can stretch really easily, and it makes sense you'd want to have that along your urinary tract where this can kind of engorge with urine. And this epithelium is also identifiable because the apical cells are larger than the basal cells, and that's typically not the case with the stratified epithelial tissues. Typically with stratified epithelia, the basal cells are larger and the apical cells get smaller. You can identify transitional because the apical cells here are much larger and pillowy versus the basal cells. And remember, these ones are good at stretch, and this only lines the ureters and urinary bladder and portions of the urethra. So think urinary tract. Now, glandular epithelia, you're going to find basically forming glands. Now, these can actually be unicellular or even multicellular. The unicellular glandular epithelial cells would be things like goblet cells. Otherwise, if they're multicellular, they're going to form larger glands like sweat glands and salivary glands. But they all make something called a secretion. And the secretion is basically, a watery solution that may contain enzymes or other molecules that give that secretion a particular characteristic. Now, it's classified based on the type of secretion it creates. Now, endocrine glands are the ones that secrete hormones in your bloodstream. Exocrine glands are the externally secreting, and these are the ones that actually put their secretion into a duct, and that duct then drains somewhere, like on the surface of your skin, or like for sweat, or along your digestive tract, for digestive juices. Now, the relative number of cells that forms the glands will tell us about whether it's unicellular or multicellular. Unicellular example of this type of gland is goblet cell, which we saw in the pseudostratified slitted columnar epithelium, or multicellular, which includes your salivary glands. So the unicellular exocrine glands are things like mucus cells or goblet cells. These you only find in epithelial linings of your intestine and respiratory tracts. They actually produce something called mucin, which is kind of a sugary protein that dissolves in water to form mucus, and it's slimy and protective and lubricates as a coating. So this is showing what the goblet cell would look like. We saw this under the microscope in the previous slides of like transitional epithelium, pseudostratified slitted columnar epithelium, as well as simple columnar. And this is the goblet cell. Its nucleus is more deep, kind of more basal. And then at the apical surface, you're going to find these large secretory vesicles that are full of mucus. And what happens is you get exocytosis of these vesicles, and it releases the mucus into the lumen or space here, whether it's your respiratory or digestive passages. Now, the multicellular exocrine glands are those that have lots of cells that are going to surround a duct. They form a secretory unit. Now, these usually are surrounded by a supportive connective tissue that supplies blood and nerve fibers. And these connective tissues form a capsule around the gland that can also extend into the gland or divide that into lobes. An example of where you find this would be like with the mammary glands for breast milk. Those are divided into distinct lobes. Now, these glandular epithelia are classified based on their structure as well as their mode of secretion. So their structure is either simple or compound. Simple means we have glands that have unbranched ducts. Compound means we have branched ducts. If it's tubular, we see that the cells form a duct, whereas alveolar, the secretory cells kind of form a larger kind of sac-shaped structure. And you can have a mixture of the two. So what this slide is showing is basically the different combinations of glands we can have. Remember, if it's tubular, it's going to be kind of more of a straight tube rather than having these large protrusions like you'd find with alveolar. Alveolar have these kind of larger sac-shaped ends where the secretory cells are more rounded. And if it's simple, that means these are not very branched, right? So it's either highly unbranched here, like just one straight duct, or it just doesn't branch a lot. So that's a simple duct. Now, compound means it has lots of branches here. So this is an example of compound. And remember, if it's tubular, we would combine these to call this a compound tubular type of gland. Or if it's alveolar, we'd call this a compound alveolar type of gland. And if it's a mixture, we just call it compound tubuloalveolar. And same thing with the simple. Remember, if it's simple but tubular, you just call it simple tubular. Or if it's simple and alveolar, you just call it simpler alveolar. Now, glandular epithelia can also be multicellular exocrine glands that can have different secretions, like merocrine secretions, holocrine, or apocrine secretions. Merocrine secretions are where you have the secretory products that are released through exocytosis. So you find this in things like sweat or pancreatic secretions. Holocrine is basically where the entire cell will accumulate the secretory product and the entire cell ruptures, and that becomes the secretion. Apocrine is basically where the apical surface of the cell ruptures, but the rest of the cell basically is maintained and intact. And this seems to be present in mammary glands. So this is showing those modes of secretion. This is an example of merocrine, where the secretory cells here would actually exocytose their secretion into the duct, and then that secretion is then transported along that duct into some sort of lumen or space. This differs from the holocrine secretion, because remember, holocrine is where the entire apical cell bursts, and the cytoplasm and all the components of that cell become the secretion itself. An example of this is sebaceous glands, which make the oil in your skin.